Have you ever noticed how the simple aroma of freshly baked bread can instantly make your mouth water?
Oh, yeah, or how a sudden, clap of thunder makes you jump.
Exactly. It's our incredible nervous system, constantly interpreting these things and building our reality.
Hmm.
But not all senses are created equal, are they? Today, we're diving deep into what scientists call the special senses.
Right. Vision, smell, taste, hearing, and equilibrium.
Yeah, and touch is considered a general sense because its receptors are everywhere. But these special ones, they have distinct, highly specialized receptors, mostly kind of tucked away right here in your head.
That's right. And for this deep dive, our mission is really to unpack the fascinating details, the anatomy and physiology behind these five senses.
We're using Human Anatomy and Physiology, the 10th edition, as our guide.
Exactly. The goal isn't just, listing facts. We want to bring clarity, maybe some surprising insights, and practical connections about these really complex systems.
Hopefully giving you those aha moments without drowning you in detail. Think of this as your shortcut, maybe, to understanding how you see, smell, taste, hear, and stay balanced. Let's jump in.
Let's do it.
So, where better to start than vision? It's clearly our dominant sense.
No question. It's staggering. About 70% of all the sensory receptors in your entire body are packed into your eyes.
70%.
Yeah. And nearly half of your brain's cerebral cortex is busy processing what you see.
Wow. Okay, before we even get inside the eyeball, let's look at the accessory structures, the things that protect it.
Good idea. Like your eyebrows. They're not just for show.
Right, they shade your eyes from bright sun, and also, maybe less glamorously, stop sweat dripping in.
Mm-hmm. And your eyelids, the palpebrae, they blink automatically every few seconds.
Protection and lubrication, right?
Exactly. And you know that whitish, oily gunk, the eye sand in the corner when you wake up?
Mm-hmm.
That comes from tiny glands there. And sometimes these glands get infected. A blocked tarsal gland can cause a chalation, like a cyst.
Or a sty, if it's one of the smaller glands.
Right, that inflammation.
We also have the conjunctiva. That's the transparent membrane lining the eyelids and covering the white part of the eye.
Produces mucus, keeps things lubricated.
Crucial. And if it gets inflamed, that's conjunctivitis.
Red, irritated eyes. Or if it's the contagious version, we call it pink eye.
Okay, so besides mucus, we have tears, produced by the lacrimal gland up in the outer corner.
Yep. It's mostly dilute saline, but it also has mucus, antibodies, and an enzyme called lysozyme.
Like a natural disinfectant.
Pretty much. Cleans and protects the eye surface with every blink.
And this explains the sniffles when you cry, right? Because tears drain into the nose.
Exactly. Tears drain through tiny ducts, the lacrimal puncta and canaliculi, into the lacrimal sac, and then down the nasolacrimal duct into your nasal cavity.
So when you have a cold and your nose is stuffed up,
Uh-huh.
that swelling can block the tear drainage, making your eyes watery.
Precisely. It all connects.
Okay, how do the eyes actually move?
6 tiny muscles, the extrinsic eye muscles, attached to the outside of each eyeball.
4 rectus muscles pull straight up, down, left, right.
And then 2 oblique muscles handle the more complex rotations, allowing for really precise movements. They work with a little pulley structure called the trochlea.
Incredible precision needed there.
Absolutely. The nerve control is extremely fine-tuned. If these muscles don't coordinate perfectly, though, you can get diplopia.
Double vision.
Right. Or if there's a weakness, often from birth, you might see strabismus, what people commonly call cross-eyed.
Okay, let's move inside the eyeball itself. 3 layers, you said.
3 distinct layers, plus the internal fluids, or humors. The outermost is the fibrous layer.
That includes the sclera, the tough white part.
Yes, the sclera. Protects, shapes the eye, anchors those muscles, and at the front, it becomes the transparent cornea.
The eye's window. And it bends light, right? A lot.
It does the most initial light bending, yes. And that cornea is amazing.
You mentioned it's packed with nerves, but has no blood vessels.
Exactly. Super sensitive to pain, heals incredibly well, and because it's avascular, corneal transplants have a very high success rate. The immune system tends to leave it alone.
Fascinating. Okay, layer 2.
The vascular layer, or uvea, rich in blood vessels, hence the name, it nourishes the eye.
It includes the colored part, the iris?
Yes. The iris, the ciliary body which controls the lens shape, and the choroid which absorbs stray light. The iris surrounds the pupil, the central opening.
And the iris controls the pupil size.
Correct. In bright light, muscles constrict the pupil. In dim light, others dilate it. It's under autonomic nervous system control.
And pupil size can show interest or emotion, too.
It can. dilates when you see something appealing or are startled. And eye color, that just depends on how much brown pigment is in your iris. Less pigment means blue or green eyes.
Interesting. And the innermost layer?
The retina, very delicate, actually develops as an outgrowth of the brain. It contains millions of photoreceptors.
Where the light detection happens.
Exactly. It has two main layers itself. An outer pigmented layer that absorbs light and helps recycle photoreceptor components, and an inner neural layer.
And the neural layer has the photoreceptors plus other neurons like bipolar cells and ganglion cells.
Yes, they process the signal. The axons of those ganglion cells form the optic nerve, which leaves the eye at the optic disc.
Ah, the blind spot.
Correct. No photoreceptors there at all. But you almost never notice it because your brain cleverly fills in the missing information using context from the surroundings and the other eye.
Smart brain. Now, photoreceptors, rods, and cones.
Two main types. Rods are incredibly sensitive to light, perfect for dim conditions and peripheral vision, but they don't do color, and the image is kind of fuzzy.
Night vision specialists.
Pretty much. Cones, on the other hand, need brighter light. They give you sharp, high-resolution color vision.
And they're concentrated somewhere specific.
Yes, mostly in a small area called the macula lutea, with the highest density right in the center, in the fovea centralis. That's where you focus for detailed tasks like reading.
Okay, what about the fluids inside? Humors, you call them.
Right. Behind the lens is the posterior segment, filled with vitreous humor. It's a clear gel, transmits light, supports the retina from the inside, and helps maintain pressure. You're born with it, and it lasts a lifetime.
And in front of the lens.
That's the anterior segment, filled with aqueous humor. It's more like a clear fluid, similar to blood plasma. It's constantly being produced and drained.
Yeah, this maintains the internal eye pressure, intraocular pressure.
Exactly. Around 16 millimeters of mercury, normally. It supports the eyeball shape and, crucially, delivers nutrients and oxygen to the lens and cornea since they don't have their own blood supply.
What happens if the drainage gets blocked?
That's a serious problem called glaucoma.
Okay.
If the aqueous humor can't drain properly, pressure builds up inside the eye.
Which damages the retina and optic nerve.
Yes, it compresses them, leading to gradual vision loss, often starting with peripheral vision and eventually blindness if untreated. It can be quite insidious, often painless initially.
So regular eye exams are key, especially after 40.
Absolutely crucial. They measure intraocular pressure routinely.
Okay, let's talk about the lens. You said it's adjustable.
Yes, the lens is transparent, biconvex, and flexible, held in place by suspensory ligaments. It can change shape to fine-tune focus.
But it changes with age.
It does. It keeps growing slowly throughout life. Adding layers like an onion, it becomes denser, larger, and critically less elastic.
Which affects focusing.
Yes, especially close-up focus. And sometimes the proteins inside, called crystallins, start to clump together.
Causing cataracts.
Exactly. A clouding of the lens. Vision becomes hazy, distorted, like looking through frosted glass. It's very common with aging, but things like diabetes, smoking, even excessive sunlight can contribute.
But it's treatable.
Oh yes, very effectively. The clouded lens is surgically removed and replaced with an artificial intraocular lens. Vision is usually restored quite well.
So how does the focusing actually work? Bending light.
Right. Refraction. Light bends when it passes from one medium to another, like from air into the cornea, then through the aqueous humor lens and vitreous humor.
The cornea does most of the bending, you said.
It provides most of the refractive power because the change from air is the biggest density jump, but the lens is the adjustable part. For distant vision, say beyond 20 feet, the light rays are nearly parallel.
So the eye doesn't need to do much.
Correct. The ciliary muscles are relaxed, the suspensory ligaments are taut, and the lens is flattened at its lowest refractive power. The eye is sort of preset for distance.
But for close vision, reading, looking at your phone.
The eye has to actively adjust. Three things happen simultaneously. First, accommodation of the lens. The ciliary muscles contract, loosening the ligaments, allowing the elastic lens to bulge, becoming more convex and increasing its bending power.
Okay, lens bulges. What else?
Second, constriction of the pupils. This prevents the most divergent light rays from the close object entering the eye, which would blur the image. It increases the depth of focus.
Like stopping down the aperture on a camera.
Exactly. And third, convergence of the eyeballs. Your eyes rotate medially, inward, so the object remains focused on the fovea of both eyes.
Which explains eye strain from close work. Those muscles are constantly working.
Precisely. Looking up and into the distance periodically lets those ciliary muscles relax.
What about common focus problems? Nearsightedness.
Myopia. or nearsightedness. Usually the eyeball is a bit too long. So distant objects focus in front of the retina, not on it. Close objects are fine, but distant ones are blurry.
Corrected with concave lenses. Or laser surgery.
Yes, those diverge the light slightly before it enters the eye. Hyperopia, farsightedness, is typically the opposite. The eyeball is too short, light focuses behind the retina.
So distant objects might be okay, but close ones are definitely blurry.
Right, especially as you age and lose accommodation, convex lenses are needed to converge the light more strongly.
And astigmatism.
That's usually due to unequal curvatures in the cornea or lens. It's like having a football-shaped cornea instead of a spherical one. Light focuses at multiple points, causing blurry vision at all distances. Corrected with specially ground cylindrical lenses or laser.
Okay, so light is focused. How does it become a signal the brain understands? Phototransduction.
Right. This happens in the rods and cones. They contain visual pigments in disc-like structures in their outer segments. These pigments absorb light photons.
Rhodopsin in rods and different ones in cones for color.
Exactly. Cones have 3 types of pigments sensitive to blue, green, and red wavelengths of light. Our perception of intermediate colors comes from stimulating combinations of these cones. If all are stimulated equally, we see white.
Which leads to color blindness, if one type of cone pigment is missing.
Yes, usually a congenital condition, often X-linked, so more common in males. Red-green color blindness is the most frequent type.
So what happens chemically when light hits a pigment?
The light-absorbing part is retinal, derived from vitamin A. It normally exists in a bent shape, 11-cis retinal. When it absorbs a photon, it straightens out to all transretinal. This shape change activates the protein part, opsin.
And this activation starts a chain reaction.
It does. It leads to what's called pigment bleaching. And here's the really counterintuitive part. In the dark, photoreceptors are actually depolarized and constantly releasing an inhibitory neurotransmitter, glutamate.
So they're on in the dark.
Effectively, yes, inhibiting the next cells. When light hits them, the pigment change triggers a cascade that closes sodium channels. This causes the cell to hyperpolarize, become more negative inside.
So light turns them off.
Right. It stops them from releasing the inhibitory glutamate.
Which then allows the next cells, the bipolar cells, to activate.
Exactly. Bipolar cells depolarize and release their excitatory neurotransmitter onto the ganglion cells.
And the ganglion cells fire action potentials that travel down the optic nerve to the brain.
That's the pathway. It seems backward, but it works.
What about adjusting to different light levels, like going outside on a sunny day?
That's light adaptation. You're initially dazzled because your supersensitive rods are bleached out almost instantly. Your cones adapt relatively quickly, though, within about 5–10 minutes, decreasing their sensitivity. Your pupils also constrict.
And going from bright light into a dark room.
Dark adaptation, much slower. Your cones stop working in the dim light, your rods have been bleached, and it takes time, maybe 20–30 minutes, for rhodopsin to regenerate and accumulate. Your retinal sensitivity gradually increases enormously.
Hence why it takes a while to see well in the dark.
Precisely. And if you lack vitamin A, you can't make enough retinol, leading to impaired rod function or night blindness.
Okay, the signal leaves the eye via the optic nerve. Where does it go?
Optic nerves from each eye meet at the optic chiasma. Here, fibers from the medial or nasal side of each retina cross over to the opposite side of the brain. Fibers from the lateral or temporal side stay on the same side.
So the left side of the brain gets input from the right half of the visual world.
Exactly. Each optic tract, after the chiasma, contains fibers from the lateral side of the eye on the same side and the medial side of the opposite eye. So the left optic tract carries a complete representation of the right half of the visual field and vice versa.
That crossing seems important.
Crucial. Most fibers then travel to the lateral geniculate nuclei of the thalamus, the brain's relay station.
And then to the visual cortex.
Yes, via the optic radiation fibers to the primary visual cortex and the occipital lobe. This is where conscious perception of visual images begins.
Does all the information go there?
Not quite. Some fibers branch off to the midbrain for visual reflexes, like coordinating head and eye movements and controlling pupil size. And some go to the hypothalamus to help regulate circadian rhythms based on light levels.
What about depth perception? Seeing in 3D?
That comes from having two eyes with overlapping visual fields. Each eye sees a slightly different view of the world. The visual cortex fuses these two images, using the differences to calculate depth and distance.
Unlike animals with eyes on the sides of their heads, who get panoramic vision but less depth.
Right, and damage along this pathway has predictable consequences. Losing an eye means losing true depth perception and peripheral vision on that side. Damage after the optic chiasma, like a stroke in the left visual cortex,
Would cause blindness in the right visual field of both eyes.
correct, because all the information from the right visual field ends up in the left cortex. Visual processing itself is hierarchical, starting with basic features like contrast and orientation in the primary cortex, and then moving to surrounding areas for interpreting form, color, and movement.
Amazing complexity. Okay, let's shift gears to the chemical senses smell and taste.
Olfaction and gustation. They're considered chemoreceptors because they respond to chemicals in solution.
And they work together closely, right?
Very much so. They complement each other and help us evaluate our environment, what's safe or desirable to eat, potential dangers.
Let's start with smell. Where are the receptors?
In the olfactory epithelium, a small patch of specialized tissue high up in the roof of your nasal cavity. That's why sniffing helps it draws more air up to that area.
And the actual receptor cells?
Our olfactory sensory neurons, they're bipolar neurons with cilia that extend into the mucus layer covering the epithelium. These cilia vastly increase the surface area for detecting odor molecules.
And these neurons get replaced. That seems unusual.
It is. They're one of the few neuron types that undergo regular turnover about every 30 to 60 days, replaced by underlying stem cells. It's quite remarkable.
How do we detect so many smells?
We have hundreds of different types of olfactory receptors encoded by a large family of smell genes. Each receptor protein binds to specific chemical features. A single odor molecule might bind to several different receptor types, and the brain interprets that unique combination, that pattern of activation, as a specific smell.
So for us to smell something,
The chemical, the odorant, has to be volatile, meaning gaseous enough to get into the nasal cavity, and then it has to dissolve in the mucus to reach the receptors.
once it binds.
It triggers a G protein mechanism, leading to the production of cyclic AMP, which opens ion channels, causing depolarization and sending a signal.
Is that why smells seem to fade if you're exposed for a while? Adaptation.
Yes, olfactory adaptation. Calcium influx during the signaling process helps inhibit the response to a sustained stimulus. You adapt pretty quickly, which is useful. Otherwise, you'd be constantly overwhelmed. Like your example of the paper mill, you notice it intensely at first, then it fades into the background.
Where do the smell signals go?
The axons of the olfactory neurons bundle together to form the olfactory nerves, which pass through the skull into the olfactory bulbs. There, they synapse with other neurons called mitral cells in structures called glomeruli.
And they?
The mitral cells send signals via the olfactory tracts mainly to the piriform lobe of the olfactory cortex for conscious awareness and identification of the smell.
But you mentioned another pathway, to the emotional brain.
Right. Some signals go directly to the hypothalamus, the amygdala, and other parts of the limbic system. This is the primitive pathway, linking smells directly to emotions, memories, and basic drives like appetite or danger avoidance. That's why a certain smell can trigger such a strong, immediate emotional response or memory.
Makes sense.
Huh.
Okay, on to taste, gustation, taste buds.
Yes, the sensory organs for taste. Most are on the tongue, located in the papillae, those little bumps you can see. But you also have some on the soft palate, inner cheeks, pharynx, even the epiglottis.
And inside each taste bud.
You have specialized gustatory epithelial cells, the actual taste receptor cells. They have long microvilli called gustatory hairs that project through a taste pore into the saliva.
And these cells get replaced too?
Yes, even faster than olfactory neurons. About every 7 to 10 days, they take a lot of wear and tear from friction and temperature changes. Basal epithelial cells act as stem cells to replace them.
What are the basic tastes we detect?
Traditionally 5. Sweet, sugars, alcohols, sour, acids, hydrogen ions, salty, metal ions like sodium chloride, bitter, alkaloids like caffeine or quinine, also many toxins, and umami.
Umami.
Yes, the savory taste, often described as meaty or brothy, elicited by amino acids like glutamate and aspartate. Think aged cheese, mushrooms, soy sauce. There's also growing evidence for a 6th taste for long-chain fatty acids.
So individual taste cells specialize.
Generally, yes. While a taste bud might contain cells sensitive to all tastes, a single gustatory cell usually has receptors for just 1 or maybe 2 taste modalities. Bitter receptors are particularly sensitive, a protective mechanism against potential poisons.
How does it work? The chemical has to dissolve.
Yes, The tastant must dissolve in saliva, diffuse into the taste pore, and contact the gustatory hairs. This binding triggers neurotransmitter release from the gustatory cell onto associated sensory nerve fibers.
And the signaling mechanisms differ for each taste.
They do. Salty and sour tastes involve direct ion channel activation, Natime plus influx for salty, H plus acting intracellularly for sour. Sweet, bitter, and umami rely on G protein-coupled effectors, specifically one called gasteusin, leading to intracellular calcium release and neurotransmitter release. Interestingly, ATP seems to act as a neurotransmitter here.
Where does the taste information go?
Signals travel via 3 cranial nerves: the facial nerve, gluteus, glossopharyngeal nerve, nicas, and vagus nerve, depending on where the taste bud is located. They converge in the medulla, then relay through the thalamus to the gustatory cortex and insula for conscious perception.
And it triggers reflexes too, like salivation.
Absolutely. Input also goes to the hypothalamus and limbic system, influencing digestive reflexes and our appreciation or dislike of foods.
You mentioned earlier that taste is heavily influenced by smell.
Hugely. Estimates say taste is maybe 80% smell. When you have a cold and your nose is blocked, food tastes bland because you're missing the olfactory component.
And texture and temperature matter too.
Definitely. Thermoreceptors, mechanoreceptors, even nociceptors, pain receptors, in the mouth contribute. Think about the difference between cold and warm pizza or the heat of chili peppers stimulating pain receptors. It all blends together for the overall flavor experience.
Fascinating how integrated it is. Okay, let's move to the ear, hearing, and balance.
Another complex organ with a dual role. Intricate machinery translating fluid movements into sound perception and our sense of orientation.
3 parts again: external, middle, internal.
Correct. The external ear consists of the auricle or pinna, the part you see which funnels sound waves into the external acoustic meatus or auditory canal.
The ear canal.
Yes, it's lined with hairs and glands that produce cerumen or earwax.
Which is protective.
Yes. Traps foreign bodies, repels insects, inhibits bacterial growth. The canal ends at the tympanic membrane.
The eardrum.
Right. A thin membrane that vibrates when sound waves hit it. This marks the boundary with the middle ear, or tympanic cavity.
An air-filled space.
Yes, within the temporal bone. It's connected to the nasopharynx by the pharyngotympanic or auditory tube.
The eustachian tube. That equalizes pressure.
Exactly the same tube. Crucial for allowing the eardrum to vibrate freely. That popping sensation when you change altitude is this tube opening to equalize pressure with the outside air.
And if it gets blocked, like with a cold?
Pressure imbalances can cause pain, muffled hearing, and can lead to otitis media, middle ear inflammation. Very common in kids because their tubes are shorter and more horizontal.
What's inside the middle ear? Those tiny bones.
The auditory ossicles, the malleus hammer, intranicus anvil, and stapes stirrup, the 3 smallest bones in the body.
And they transmit the vibrations.
Yes, they form a lever system. The malleus is attached to the eardrum. The stapes fits into the oval window, a membrane leading to the internal ear. They transmit the eardrum's vibrations and, importantly, amplify the pressure.
Amplify. Why is that needed?
Because they're transferring vibrations from air in the middle ear to fluid in the internal ear, which is much harder to move. The ossicles increase the pressure about 20-fold, mostly because the eardrum is much larger than the oval window.
Clever mechanics. Are there muscles there too?
Tiny ones. The tensor tympani and stupedius. They contract reflexively in response to loud sounds, dampening the vibrations to protect the delicate internal ear structures.
Okay, the internal ear, the labyrinth.
Yes, deep in the temporal bone. It has 2 parts. A bony labyrinth, which is a system of channels filled with a fluid called perilymph, and suspended within that, a membranous labyrinth, a series of sacs and ducts filled with endolymph.
And 3 main regions within the bony labyrinth.
Right. The vestibule is the central cavity containing sacs called the saccule and utricle. These house maculae, the equilibrium receptors for gravity and linear acceleration.
Like starting and stopping.
Exactly. Then there are the 3 semicircular canals, anterior, posterior, and lateral oriented in the 3 planes of space. Each has a swelling called an ampulla at one end, housing a crista ampullaris, the equilibrium receptor for rotational movements.
Spinning and turning. And the 3rd region.
The cochlea, snail-shaped. This is purely for hearing. It contains the cochlear duct or scala media, which houses the spiral organ, also called the organ of Corti. This is the actual receptor organ for hearing.
So hearing is basically vibrations traveling through all these parts.
It's a chain reaction. Sound waves in air, eardrum vibration, ossicle vibration, pressure waves in the parallel fluid vibration membranes within the cochlea, bending of hair cells in the spiral organ, generation of nerve impulses, interpretation by the brain.
Let's unpack sound itself, frequency and amplitude.
Sound is a pressure disturbance. Its frequency, the number of waves per second, determines its pitch. Higher frequency, higher pitch. Measured in hertz, its amplitude or intensity determines its loudness. Measured in decibels, dB.
And loud sounds can be damaging.
Absolutely. Prolonged exposure to sounds above 90 dB can cause permanent hearing loss.
So how does the cochlea sort out different pitches?
Through the basilar membrane, which supports the spiral organ. This membrane's properties change along its length. Near the oval window, the base, it's narrow and stiff, resonating with high-frequency sounds. Near the far end, the apex, it's wider and more flexible, resonating with low-frequency sounds.
Like a piano strings.
A very good analogy. Different frequencies cause maximal vibration at different points along the basilar membrane. This mechanical frequency analysis is key to pitch perception.
And this vibration moves the hair cells.
Yes. The spiral organ sits on the basilar membrane. As the membrane vibrates, the stereocilia, the hairs on the inner hair cells, are bent against an overlying membrane called the tectorial membrane.
And the bending opens channels.
It does. Bending the stereocilia towards the tallest one stretches connecting filaments called tip links, which mechanically pull open ion channels. Positively charged ions, mostly potassium K+, and calcium Ca2+, flood in from the surrounding endolymph.
Wait, potassium causes depolarization. Isn't usually sodium?
In most neurons, yes, but the endolymph fluid in the cochlear duct is unusually rich in K+. So K+ influx depolarizes the hair cell. This causes it to release neurotransmitter, glutamate, exciting the auditory nerve fibers.
And bending the other way.
Bending away from the tallest cilium closes the channels, hyperpolarizing the cell and reducing neurotransmitter release.
So the inner hair cells send the signals. What about the outer hair cells? There are more of them.
Their role is fascinating and was mysterious for a long time. They receive mostly efferent signals from the brain. When stimulated, they actively change length. They contract and elongate very rapidly. They literally boogie.
Boogie. What does that do?
This electromotility changes the stiffness of the basilar membrane locally. It amplifies the motion of the membrane in response to soft sounds, effectively tuning the cochlea, making the inner hair cells much more sensitive. It also helps protect the inner hair cells from loud sounds.
Wow, active amplification. Okay, where do the auditory signals go in the brain?
From the cochlear nerve, they travel through several processing stations in the brainstem, cochlear nuclei, superior olivary nucleus, then to the inferior colliculus in the midbrain, then the medial geniculate nucleus of the thalamus, and finally to the primary auditory cortex in the temporal lobe.
And processing happens along the way.
Yes. Pitch is perceived based on which hair cells along the basilar membrane are activated. Loudness is detected by how much the basilar membrane moves, leading to more frequent action potentials.
How do we know where a sound is coming from?
Sound localization uses two main cues: the difference in intensity, loudness, and the difference in the timing of the sound waves reaching each ear. The brain calculates these tiny differences to pinpoint the source.
Incredible. Now, let's switch to the other function of the ear balance, or equilibrium.
Right, the vestibular system. It works largely unconsciously, integrating input from the internal ear, vision, and proprioceptors, stretch receptors in muscles and joints, to keep us upright and coordinated.
Two main parts in the inner ear for this, the maculae and the cristae.
Exactly. The maculae, located in the saccule and utricle of the vestibule, are responsible for static equilibrium. They monitor the position of your head relative to gravity and respond to linear acceleration, straight line changes in speed or direction.
How do they work?
They have hair cells whose stereocilia are embedded in an overlying gelatinous layer called the otolith membrane. This membrane contains tiny calcium carbonate crystals called otoliths, ear stones.
Ear stones.
Yes. They add weight and inertia. When you tilt your head or accelerate linearly, like in a car starting or stopping, gravity pulls on the otolith membrane, or inertia makes it lag behind. This bends the hair cells, signaling the change in head position or linear motion.
Clever use of physics.
Okay.
And the cristae.
The cristae ampullaires are located in the ampullae of the semicircular canals. They respond to dynamic equilibrium, specifically rotational or angular movements of the head.
Like shaking your head no or nodding yes.
Precisely. Each crista has hair cells whose stereocilia are embedded in a gel-like cap called the ampullary cupula. When you rotate your head, the endolymph fluid inside the semicircular ducts lags behind due to inertia. This fluid pushes against the cupula, bending the hair cells.
Signaling rotation.
Yes. Because the three canals are oriented in different planes, combinations of signals from them tell the brain exactly how your head is rotating.
What happens if you spin at a constant rate?
Interesting effect. After a few seconds, the endolymph catches up and starts moving at the same speed as your head. The cupula is no longer bent, so the stimulation stops. If you were blindfolded, you wouldn't be able to tell you were still rotating at a constant velocity.
Until you stop.
Right. When you stop suddenly, the fluid keeps going for a moment due to inertia, bending the cupula in the opposite direction. This signals deceleration and can cause that feeling of dizziness or vertigo, and often jerky eye movements called vestibular nystagmus.
Where does all this balance information go? Does it reach conscious awareness?
Most of it doesn't. Equilibrium signals primarily go to reflex centers in the brainstem, the vestibular nuclei, and to the cerebellum. These areas use the information to rapidly adjust muscle activity to maintain balance and control eye movements to keep your gaze fixed even when your head is moving. The vestibulo-ocular reflex.
Which explains why balance feels so automatic.
Exactly. It's mostly reflexive.
What about motion sickness then?
That seems to happen when your brain receives conflicting sensory information about motion. For example, on a boat in rough seas, your eyes might see the stable cabin interior, telling your brain you're stationary. But your inner ear's vestibular system is detecting the boat's pitching and rolling. This mismatch confuses the brain, often leading to symptoms like nausea, dizziness, and vomiting.
Makes sense. Okay, let's wrap up with how these senses develop and what can go wrong.
Sure. Taste and smell are actually sharpest at birth. Babies have a wider appreciation for different tastes than adults sometimes do, but these senses tend to decline with age, partly because receptor cell replacement slows down. Over half of people over 65 have significant issues, which can affect appetite and nutrition.
What about vision? Fully formed at birth?
Not at all. Vision is probably the least mature sense at birth. Newborns are hyperopic, farsighted, see mostly gray tones, lack coordinated eye movements, and don't produce tears for the first couple of weeks.
When does it get better?
Depth perception and color vision develop gradually, usually well established by age 3. The eye continues to grow, reaching adult size around age 8 or 9.
And age-related changes later in life.
Common ones include presbyopia, the loss of lens elasticity around age 40, making close vision difficult. The lens may also become less transparent, cataracts. Pupil size might decrease, and overall visual acuity often declines in the elderly. Certain congenital issues can also arise, like blindness or cataracts if the mother had rubella during pregnancy.
And hearing development.
The ear starts developing very early in the embryo. Newborns can hear, though responses are initially reflexive. By about 4 months, they'll turn towards voices. This early auditory input is critical for language development.
Does hearing also decline with age?
Yes, presbycusis, or age-related sensorineural hearing loss, is very common. It typically affects high-pitched sounds first. Worryingly, it's becoming more common in younger people too, likely due to increased exposure to loud noise concerts, personal music players.
So it's often due to hair cell damage.
Yes, gradual damage or destruction of cochlear hair cells from noise, certain diseases, or ototoxic drugs is a major cause of sensorineural deafness.
How is deafness classified?
2 main types. Conduction deafness means something is blocking sound transmission in the external or middle ear, could be impacted earwax, a perforated eardrum, otitis media, or otosclerosis, where the auditory ossicles fuse together. Hearing aids often help hear by amplifying sound. And the other type.
Sensorineural deafness. This involves damage to the neural structures anywhere from the hair cells to the auditory cortex. This is usually caused by hair cell loss. While hearing aids might help somewhat, for severe cases, cochlear implants can be revolutionary. They bypass the damaged hair cells and directly stimulate the auditory nerve.
A remarkable technology.
What about ringing in the ears, tinnitus?
Tinnitus is the perception of sound ringing, clicking, buzzing in the absence of any actual external sound. It's a symptom, not a disease itself, often indicating degeneration or inflammation of the auditory nerve pathway, or sometimes side effects from drugs like aspirin. It can be quite distressing.
And finally, Meniere's syndrome.
That's a disorder affecting the entire labyrinth of the internal ear. It causes recurrent episodes of severe vertigo, nausea, vomiting, tinnitus, and fluctuating hearing loss. It's thought to be related to an excessive accumulation of endolymph fluid, distorting the membranous labyrinth.
What an incredible journey we've taken through our special senses. From the intricate mechanics of the eye-focusing light,
To the microscopic wonders of smell and taste receptors detecting chemicals.
and the amazing fluid dynamics governing hearing and balance. It's just so clear how complex and interconnected these systems are.
Absolutely. Constantly working together, often unconsciously, to shape our perception of the world around us.
It really is a remarkable biological choreography.
It truly is. And maybe a final thought for you listening. Consider how our brains aren't just passively receiving this information. They actively construct our reality from these sensory inputs, constantly filling in blanks, blending signals, making predictions. What stands out to you as the most surprising part of this whole intricate process?
Thank you so much for being part of this deep dive with us and for your curiosity.